All right, so now we're going to move on to how smooth muscles contract for the main focus of this lecture, along with muscle twitches and fast twitch and slow twitch. Those are the kind of things that we're going to talk about today. When you watch this presentation, realize that you need to write a summary of what you learned as well with, I will pick a figure for you to diagram, which will be on smooth muscles. I believe this is lecture number 3 on the skeletal muscles and smooth muscles in the animal physiology series. So it's either lecture 3 or lecture 4, but I'm pretty sure it's lecture 3 today. And so let's go on with whatever I label will be the correct one, but let's go ahead and start with kind of reminding ourselves what happened with skeletal muscles contraction. And we're going to use that to compare it with the smooth muscle. Knowing how excitation contraction coupling works in skeletal muscle is key to this portion of the class. You really need to understand this. So this is going to be heavily tested for. And so just again, looking at this diagram, you need to be able to draw it and diagram it and explain it on your own without referring to a book. So if you look at this, this is our axon. And over here it says the somatic motor neuron releases acetylcholine as a neuromuscular junction. So we talked about this before. But so you see the neurotransmitters coming across, and this triggers sodium to come in, and we call that depolarization or action potential. As you remember, that travels down the T tubules, opening up a DHP, or triggering the DHP receptor to open up a channel on the sarcoplasmic reticulum to allow a bunch of calcium to enter, attach to tropomyosin, and move the troponin out of the way that allows for the cross bridges. And then if you follow that, if you remember a couple of lectures ago, following the comic book strip, you can see that the myosin grabs on and uses ATP and breaks it down to do the power stroke and brings the sarcomeres together. So again, the sodium, so the acetylcholine receptors allows the entry of sodium. Now it's interesting that skeletal muscle, so it's really complete as regulated by interacting with nerves. Smooth muscle, on the other hand, may be nerves, but it's also can be stretch receptors stretching the smooth muscle, allowing calcium to enter. So it's a little bit different. Also, as you'll see, there is less sarcoplasmic reticulum in smooth muscles. Remember, skeletal muscles have a lot of nuclei, hundreds of nuclei, while smooth muscles have one nucleus and elliptical. You don't see the striations. So if you go to the next, going back to the skeletal muscle on excitation, contraction coupling, remember excitation is the nerve exciting the cell membrane. And so here's the calcium being released, as I mentioned in the previous slide. So the action potential in the T-tubule alters conformational changes, molecular changes to the DHP receptor that ultimately opens up the calcium to be released from the sarcoplasmic reticulum. So it opens up a calcium channel. And then calcium binds the troponin. And again, as I mentioned, it moves the tropomyosin out of the way, and then we got the myosin heads doing its thing. So that's kind of the framework. Now realize this is skeletal muscle. Now let's see what's happening with smooth muscle. We've already talked about this and how it all happens within milliseconds. That was in our previous lectures. Now here's the smooth muscle. Notice, again, one nucleus, even though I don't see it in this picture. You see that you don't have sarcomeres all nicely lined up, causing the formation of striations. Instead, we see these dense bodies, and this light purple that you see in this picture is representing the actin. And down here, you can see the zoom picture. If you look at my cursor, you can see the actin filaments. We also call it the thin filaments. And then you'll see the green in this picture represents the myosin, and down here we see the myosin heads and the thick filaments. And then we have the dense bodies that hold the actin. But instead of being all kind of linear, it's going in different directions. And I think as I showed you, in the previous lecture, it causes the smooth muscles to close and cramp up, ultimately allowing like a lumen of your intestine to shrink and help to churn the food and move it forward as it goes through the small intestine or the stomach or whatnot. Also remember there's smooth muscles even around the eye for dilation, the pupil, and so forth. So again, we don't see sarcomeres in regards to smooth muscle. There are gap junctions often, depending on what type of smooth muscle arrangement you have. You have some smooth muscles that will be connected through these gap junctions, which is like a channel or door that connects two cells together. So this is a cell, and this is a cell, and would be like an attachment that allows them to talk to each other. That is a gap junction. And but not all smooth muscles have that. Some of them stay in isolation, relatively speaking. And again, they can contract and stretch tremendously. There's a lot more actin with smooth muscle than there is with skeletal muscle in comparison to myosin. So there's 16 times more actin with smooth muscles in relationship to myosin, while in skeletal muscles, the myosin is much more prevalent. And again, there's these dense bodies that are the anchoring bodies for the thin filaments. So this couple descriptions will make a little bit more sense when you see the next picture. Remember, calcium was critical for having skeletal muscles contract by the calcium attaching to troponin and moving tropomyosin out of the way. So this is really important. There's no troponin or tropomyosin in the smooth muscles, so that is not being utilized. Instead, there's a protein called calmulin that is kind of like an on and off switch. It's basically a protein that, even if you're familiar with chemical cascades, it's a protein where the calcium will attach to, and then like a light switch, it'll flip, and then activate what we call myosin light chain kinase. Again, a picture will help with this, but essentially what that does is it will put a phosphate or phosphorylate myosin. That means you think about ATP and the phosphates, and that allow the phosphate or the myosin to do its thing. Now, it may sound a little bit confusing. You'll see a picture, and that diagram should hopefully help you to understand it, but basically it allows the myosin to grab onto actin and then do its contractions. This slide is basically saying the same thing. In smooth muscle, calcium is entering the sarcoplasmic reticulum and combines with the protein calmulin. Now remember, the sarcoplasm is like part of the endoplasmic reticulum. That's why I kept saying reticulum. Sarcoplasma holds the calcium. Now, in skeletal muscle, it's a huge amount. There's a lot of it. It's very specialized. But in smooth muscle, the sarcoplasm is much, much, much smaller. Also, calcium can come in extracellularly. That means the calcium can come in from the outside entrance of the cell. And again, the diagram will help with this. The calmulin, again, is just an on-off switch that will activate a myosin kinase And then that will phosphorylate the myosin heads. Remember the myosin, the heads? It's like the little hands that grab onto the actin. And from there, we'll have the contraction. Again, there's no troponin or tropomyosin in regards to the smooth muscle. So here's the diagram that you should be familiar with in regards to smooth muscle. You can see that we have the extracellular fluid, that's what ECF represents. And then calcium comes in through a calcium channel. This may be due to stretching on the receptor, or maybe some nerves influencing it, like with acetylcholine and so forth. Here we do have a sarcoplasmic reticulum, but it's a lot smaller in smooth muscle, and it causes the release of calcium as well. And then in this diagram, you see that this is a protein, calmulin, as we discussed in the previous slides, where calcium comes along and by its action on an attachment to calmulin, it causes the calmulin to release a phosphate that will activate an inactive myosin light kinase. So an inactive myosin light kinase will be activated by the release of a phosphate. So then this enzyme becomes, or this protein becomes activated, enzyme protein becomes activated, where ATP can then phosphorylate the myosin heads, adding the phosphate. Now remember, we saw something like this in a skeletal muscle slide where an ATP becomes ADP in a free phosphate. And this will allow the myosin to grab onto the actin and do its contraction. So there's no troponin or tropomyosin involved in this. So again, the calcium calmulin complex will activate this other enzyme called myosin light chain kinase, MLCK. And it's through this phosphorylation that the myosin will utilize ATPase. Remember what ATPase does? That breaks down ATP, androgen triphosphate, to androgen diphosphate, giving the myosin its ability, because remember, where do we see the ATPase? On the myosin, and that will allow it to break down, or so at least it's closely associated with the myosin, to break down androgen triphosphate to androgen diphosphate, allowing it to attach to the actin and increase muscle tension. And so after the after the myosin grabs onto the actin with its phosphorylation, it begins to move the actin and do it in a way that causes the filaments to pass past each other. Remember, in skeletal muscles, the sarcomeres came together. Well, in the case of smooth muscles, they run past each other in such a way that it causes the entire smooth muscle to contract. So look at this picture here. Remember what the dense bodies do? They grab onto the actin and and anchor the actin. Remember, in cells have cytoskeleton. So this dense body is attached to some type of cytoskeleton, and then the thick myosin the thick myosin is pushing the actin across it, so it actually goes past each other. So it's going this way and this way, and not just in a nice sarcomere going back and forth. So it actually can go past each other and keep going to make a very tight contraction, as you can see in this picture up here where we have A going into B, forming this globular shape that really shrinks a lumen. So remember, smooth muscles are usually surrounding like and this is representing a tube, the lumen, and we can shrink it during a contraction for like intestines and so forth. So that's what you're seeing in this picture. Now, how does this relax? Well, eventually the calcium is pumped back out, and then the myosin is dephosphorylated. So relaxation occurs when the calcium concentration decreases and the myosin is dephosphorylated by myosin phosphatase. Myosin can no longer form these cross bridges, and so the smooth muscle begins to relax, and it gets back to its relaxed state. So we go from a crunch state to a relaxed state. And so that's what's shown in this picture here. The myosin phosphatase becomes reverses and makes ATP again. The muscles relax down here. The calmulin releases its calcium, becomes calmulin by itself, and then that free calcium comes up and gets pumped back into the small sarcoplasmic reticulum of smooth muscles, or it goes into this cavula, or it gets pumped out of the cell through the use of ATP, because it's again going from it, because out here the calcium would be at a higher concentration. So calcium gets pumped out, sodium gets pumped in. So ultimately what happens is the cytosol, which is the watery area of cells, free calcium in the cytosol decreases, and the calcium is pumped out of the cell or back into the sarcoplasmic reticulum. The calcium unbinds from calmulin. The myosin phosphatase removes the phosphate from myosin and thus decreasing ATPase activity. Thus, myosin ATPase results in decreased muscle tension. So let's take a moment and see if I can show maybe an animation on this. Okay, so I'm just going to go ahead and continue. I will provide a couple of videos on smooth muscle contraction in your homework assignments, just because I don't see anything that looks really appropriate to add to this particular lecture. That wouldn't just be its own lecture. So let's talk about the fact that, let's go back to skeletal muscles now and talk a little bit about muscles in regards to strength and performance. Skeletal muscles come in different varieties. Some of them are what we call slow twitch fibers. They tend to be dark red in color, and they have more mitochondria associated with it. These tend to be muscles that have a lot of endurance in regards to skeletal muscles. So these are the muscles that help you stand up all day and walk long distances. They also have a lot of myoglobin associated with them. Now, if you remember the term hemoglobin, hemoglobin is this protein that's found in oxygen, or excuse me, holds on to oxygen. It's found in red blood cells. So hemoglobin is a protein that has iron units, and oxygen will grab onto it and give blood its red color. Well, myoglobin is a lot like hemoglobin, except it's found in muscles, and it takes the oxygen from the hemoglobin when it's traveling through capillaries by the muscles and helps give muscle its red color. While red or slow twitch fibers also tend to be darker and red, are these muscles, and again, just like the name says, they're just a little bit slower in their contraction rate, but they have the ability to have a lot of endurance. Why is having mitochondria associated with slow twitch fibers so important is it also contributes to its endurance. Now, so again, you're going to see a lot of glycogen and fat that goes along with it, because fat's good for more often twitches. Now, what about so where do we see these kind of muscles tend to be geared towards longer active activities like swimming, running, and so forth like that, if it's not sprint related. Now, fast twitch, the muscle fibers actually have a more white appearance. And again, I'll show you a few pictures in a moment. They have fewer mitochondria, and they have little myoglobin. They actually wear out very quickly, but they're very strong and large in size and diameter in comparison to smooth muscles. They have lots of ATPases associated with it, so that allows for ATP to be broken down and for that quick power stroke. that'll be needed for sprinting, lifting heavy weights, and so forth. Fast twitch fibers tend to be ideal for sudden, rapid weightlifting or sprinting. However, a lot of this is genetically largely determined based on the proportion that you have of these two different fibers. So somebody with a high vertical jump might naturally be gifted, if that's what we care most about, towards sprinting and throwing and jumping. While those with slow twitch fibers tend to be more gifted towards long distance sports. But again, you'll actually see a slight difference in color. It'll be, we call it white muscle, but it's actually a light pink in color in comparison to smooth muscles, which is dark red in color. Why is it whiter in color? There's less myoglobin, because remember, myoglobin is like hemoglobin. And so that iron and the oxygen combined give it that dark red color. There's a lot less mitochondria associated with it. So again, we have these three different types of muscle fibers. We have the ultra fast twitch fibers called glycolytic fast twitch, which are fast and they're anaerobic. That means they don't utilize oxygen. The downside of having all this power is they wear out very rapidly. And then we have what we call oxidative fast twitch, which is kind of an intermediate twitch, because again, when we say twitch, we're actually talking about how fast the twitch occurs. So a fast twitch would be within a little bit of a millisecond, while slow twitch might be just a little longer. So oxidative fast twitch is somewhere in the middle. And it uses a combination of anaerobic without oxygen. So sprinting is an example of anaerobic sport. Weightlifting is another example of anaerobic sport, while slow-paced long-distance running is aerobic, utilizing oxygen. So this oxidative fast twitch kind of is the intermediate between these extremes, where you have the glycolytic fast twitch, and then you have the slow twitch is very slow, much slower at fatiguing. So a lot more mitochondria, a lot more capillaries, a lot more myoglobin. Excellent for posterior and endurance activities. Now, if you ever met anybody that's really extremely gifted in fast twitch fibers, they're less gifted when it comes to endurance. I've seen some great sprinters that have a hard time running a couple of miles, but they're great at that 40-yard dash or 100-meter dash. But they're awful. They couldn't even begin to think about really running a marathon and being successful and competing. So this is why you don't see athletes really doing all these different things. Now, somebody with oxidative fast twitch fibers predominantly might make a good decathlete because they're able to do endurance and sprinting activities and shot put and things like that. But the extremes don't seem to do very well. And a lot of times, a lot of that is due to genetics, some of that's due to training. But the proportion of these muscle fiber types you have is something that you're genetically inherited with. So here's a picture showing you slow twitch fibers versus fast twitch fibers. You can see the slow twitch fibers have smaller capillaries, they look smaller diameter, but large capillaries in relationship to it. These spaces right here is the capillaries, as you can see. Remember, capillaries are little small blood vessels that blood travels through that helps to oxygenate the red muscle, the slow twitch fibers. So why are they? When they're slow twitch and they're smaller in diameter, that means there's a lot less of the myofibrils. Remember all the myofibrils with the sacromeres and things like that? There are less of them. But also, being smaller diameter, substances can enter and exit the cell more easily. Now contrast that with the fast twitch fibers, which again, it's going to be a very quick twitch. If you were to measure the speed at which the muscle's moving, the fast twitch fibers would have a much stronger contraction, allow you to lift heavier weights, sprint faster, but they fatigue incredibly quick in comparison. And so you can see how large the diameters of these what they call fast twitch or white muscle fibers. Again, they're not white, but they're light pink because they have a lot less myoglobin. And while the dark red has a lot more myoglobin. So looking at another diagram, you can see that a person doesn't have all fast twitch or all slow twitch. It can actually be intermingled. So here's the diagram above. There's more slow twitch to more slow twitch to fast twitch fibers. While this muscle biopsy, when we can take a little needle and measure the muscles and look at them under a microscope, you see there's a lot more fast twitch to slow twitch. So when you look at the relative percentage of slow twitch muscle fibers to fast twitch, you can see that some athletes have a lot more fast twitch fibers, like sprinters, for instance. But again, they're going to do horrible at really long distance running. Weightlifters are going to have a tough time too, but they can lift some massive weights. Cross-country skiers, on the other end, have a lot more slow twitch fibers, and they can go all day. And long-distance runners can do that too. Now, swimmers are somewhere in the middle, and that makes sense. And a lot of swimming does involve a certain amount of sprinting and lots of endurance. So anyhow, that kind of gives you some idea about slow twitch fibers and fast twitch fibers. And again, remember there's two different, and there's an intermediate fiber, which is more geared towards being a fast twitch. So again, how does muscle contraction occur? At least in regards to skeletal muscle, it's initiated by action potentials that travel down the motor neuron, as I described before. We call these motor units when neurons can talk to more than one muscle fiber and cause it to activate. What can activate or excite these muscle fibers? Again, it's going to be they're excitable by the acetylcholine depolarizing the cell membrane and causing sodium to enter. The minimum unit of contraction is what we call a twitch. So literally just how fast, you can actually see this, and you'll see this when you do your physiograph. A twitch is just a little bit of a contraction. And the tension it generates a twitch. Again, this tends to be very rapid. So like if I was to do electrical shock on a frog's arm, you would see a twitch. And if I was to give it lots of shocks, you'll see more twitches going. And you'll see this in another diagram. And then if I was to give it a lot of shocks, you would call it tetanus, and then all the muscles are contracting really hard. And it takes longer for the calcium to be removed between when you're just having calcium constantly being released. That's when the muscle goes into tetanus. So this is a diagram that you'll see from using a physiograph, and you'll get to try this on your homework from the physiograph lab simulations. Here is what we see. We see this represents electrical stimulus, and you'll see that it shocks the muscle and causes a twitch that relaxes. And then you'll see that was an individual twitch. And then you'll see another twitch. But what happens if we did the two electrical stimulations at once, or within closer proximity in time? So this is far apart at the beginning, and we saw a twitch. And if you look at my cursor, we see two electrical stimuli closer together than over here. Well, what we have is called summation, where the muscle twitch actually increases in height. So the contraction becomes even stronger because the twitches took place more rapidly and the calcium has not left, allowing for more myosin to grab a hold of the actin. And so down here, we see another diagram where we have different electrical stimuli being inputted. And you can see a summation taking place because we have them relatively close together. But notice what happens when we do lots of electrical shocks. Now, let me step back a little bit. You might be what am I talking about? Imagine doing an experiment that you'll get to do from your lab simulation where you had a frog leg hooked up to a machine that can measure how much contraction takes place. And so you're shocking the frog leg and maybe turning on the computer and telling it to give it one shock, and you see this tension, you see two shocks to give it, and it makes a stronger tension. That's what this picture diagram is demonstrating. And so looking at this, you can see the summation where you have electrical shocks that are close to each other, increasing the amount of contraction. Now here we have lots of electrical shocks, and all of a sudden we have a very strong contraction. And then when we stop shocking the frog legs muscle or your own muscle, it doesn't really matter, you'll see the muscles begin to relax and the tension or the force is a lot weaker. And so that's what this side is showing you, force. And this is the electrical stimuli. So the length of time that the electrical shock takes place in tetanus depends on the supply of ATP. Once that ATP disappears, the muscle is going to begin to relax also. But if you have lots of twitches occurring faster, we have this summation. We call this a temporal summation if it's done rapidly in time. But spatial summation, if more than one nerve is coming in and influencing a muscle fiber, that would be called spatial summation. Okay, so looking at this diagram here, this is just another picture showing you what happens if a frog leg is attached. And then if you give it electrical shock, you see single twitches. That's pulling, imagine a string pulling down on this machine that will measure. And if this is confusing, you'll get to do the lab assignment, and then this will make more sense when you come back to it. And then if you have electrical shock, it's more, and this is just like the previous diagram, in rapid succession, we call it spatial, or we call that summation. So we have a stronger contraction. See how the force goes up? So if you shock the frog leg a little bit more rapidly, we get a stronger summation. If this is a little bit confusing, again, you will make more sense when you do the lab. And I will see if I have a video that can help with this as well. And so here's summation that leads to what we call tetanus, and in this case, unfused tetanus. So the physiograph machine can still pick up on the individual tension and the force gets a little bit stronger. While over here, this picture is depicting, again, a muscle contraction that we're able to measure. And if you shock the heck out of it, see all the electrical shocks that take place over here and over here, the muscle is just going to go into strong, or what they call complete tetanus. And you won't be able to tell the difference. You see the individual contractions. Imagine my arm being the frogs. Over here we have unfused tetanus, where my arm's flexing. And then over here we have complete tetanus, and you can't no longer see because it's so tight. It's like lifting a heavy weight at that point. Now, what you got to keep in mind that muscles don't have to do that much contraction for the lever to move quite a bit. And so we have a fulcrum here where their elbow, and this is true for any single joint. And so, and then the muscle has different attachments. And I'm not going to go into a lot of detail about this, but if the muscle is attached further out, the distance that you move your arm is going to be shorter. And so it could actually make you have a little bit stronger. So there is something to leverages. Different people have different leverages that might make them a little stronger. If the leverage is a little closer, they'll tend to be a little weaker, but maybe they can move their arm further and have a stronger throwing power or something of that nature. And so it doesn't take much movement on this fulcrum, maybe 1 centimeter, and I'll move the hand 5 centimeters. And so you can see how the fulcrums are vary based on the different joints. Here we got a knee where the hamstring's attached. And a little bit of a 1-inch move causes 5 inches of move on the leg. While muscles that are more directly attached, like in the case of the jaw, it's almost a 1-to-2 ratio. So when you move your muscle a little bit, it's really moving your jaw. Or not moving your jaw very far, but And then these tend to be, so these tend to be faster and these tend to be stronger. Now, you might be familiar with isometric exercises and isotonic exercises. Isotonic exercises is when your muscles are strong enough to lift the weight and set it down. That's isotonic. The muscles have contracted. Isometric is when the weight is too hard that you can't lift it. It's like pushing against this wall. You cannot. That would be isometric exercises. So the muscles aren't really shrinking up. Now, there are some fibers still contracting, because you know when you do this, you can feel the muscles contracting, but the weight is too heavy to move. We call that isometric exercises. And so the muscle can move a little bit. The myosin and actin will still move a little bit. But what allows for that is there's some stretch ligaments and things like that that allow for the contraction to occur, even though the arm isn't able to move its distance. And so here is an example of what's happening in the arm. This is the muscles are relaxed. We have this elastic fiber, we have the myosin, and we have our Z lines. And so this would represent a sarcomere. In an isotonic contraction, where you're able to lift the weight, the muscles contract and the sarcomeres come closer together. But in isometric, you lift and try to push, pull, and you can't bring the sarcomeres together for the most part. Just a little bit, but the elastic fibers allow for the muscles to make some form of contraction. Lastly, I want to talk about where does your sarcomeres normally look like when they're relaxed? And that is in this picture here. In this picture, this is what the normal relaxing state of your sarcomeres look like in regards to the myosin and the actin. There's a little bit of overlap with the myosin and the actin, and here's our Z lines. And so this is where muscles are mostly located, the sarcomeres, where the sarcomeres are contractile unit. It's just kind of this middle ground where some of the myosin is attached to some of the actin or next to it. And so when the muscle is triggered into contraction, this is where you get the strongest muscle contraction. Now, if your muscles are all cramped up and the sarcomeres come closer together, you end up in a state where your muscles are no longer very strong because they've already contracted. There's no room left for the sarcomere to come together. And so if you're to trigger this muscle that's already contracted, you won't see any more relative tension or strength. Now, let's say you've overstretched your muscles. Now, if you look at this right diagram, the myosin and the actin are no longer overlapping very well. There's just a little bit of myosin here. And then when the muscle begins to contract, it's actually weaker. So overstretching your muscles before a workout actually can make your muscles a little bit weaker when you contract, from what I understand. So the optimal space is this kind of relaxed space here. So this is the relationship between the myosin and the actin in regards to overall strength when the muscles contract. If it's overstretched, the muscles are pretty weak. If it's too close together, the muscles are weak. It's this happy medium where the muscles will have the greatest strength to move. But just remember, even when your arms are just relaxed and you're not doing much of anything, you still have some muscle tone. Some muscles are contracting a little bit at any given moment, and we call that muscle tone. And that's due to the nerves providing their impulses. And so they're providing a little bit of contraction on these different fibers, even when you're in a relaxed state. So that ends most of my muscle lecture. If you go back to the very beginning slide, you will see the suggestions that you need to prepare for the study guide. Notice that right here is actually a question. So let me fix that real quick. So this is your study guide for an upcoming exam that you will have for animal physiology. These are questions that you will need to be able to answer as exam questions. So you will need to practice it, and they will become homework questions. You can look them up online, or you can look them up online, or you may, like this one, hyperkalemia. I haven't talked about that. You'll need to look it up. I may provide a little bit more lecture on these two here. But this is the essay portion of the exam. I haven't quite figured out how I'm going to do it yet. But this is the exam that you will be doing for the class. This is the study guide. I'm going to go ahead and save it. So take a note of this, look at it, make sure you realize I fixed this one here. So what do you need to do for this lecture? I want you to write a summary of how smooth muscles contract, and I want you to diagram smooth muscle contraction, which would be slide 67. So write a half a page, single space, type it, and draw this picture. Don't just take a picture of it, draw the picture and diagram it if you want full credit for attendance for this lecture. I will get back with you with some additional details as we figure out how we're going to do the exam and so forth. So that concludes this lecture. Take care.